Hi, welcome to video 2 of this lesson where we're going to talk a little bit about the specifics of how actin and myosin cause contraction in skeletal muscle. Again, you'll find that this is very similar to cardiac muscle with just one or two differences. First of all, here is our myosin molecule, which makes up our heavy filament. These filaments sort of wrap around each other and make what looks like a bunch of golf club heads sticking out occasionally in this bundle of myosin fibers, okay? And these myosin fibers have heads or cross bridges, and on those heads you actually have two binding sites. You have a actin binding site, which binds actin, the thin filament. Naturally, when you get contraction, you have a myosin ATPase site, which binds and hydrolyzes ATP and uses that energy to cause contraction. And these two binding sites work together in a way that I'll explain again or review with you again in a moment. Okay, the thin filament made up of actin, tropomyosin, and troponin. The actin monomers, or what you see here, they have a myosin binding site. So a binding site where you get attachment to the myosin and where contraction takes place. And these will actually are assembled into what looks like a string of pearls here, with the actin myosin binding sites sort of like sticking out and exposed. Wrapped around the actin is tropomyosin and troponin. So the tropomyosin wraps around, and in skeletal muscle at rest, which is meaning not contracting, this tropomyosin covers these actin binding sites, okay? And the troponin, there are three types of troponin, and they act to connect this tropomyosin to the actin, and of course, they are the key to initiation of contraction, as you will recall. So if we look at the arrangement of the actin and myosin, this is an electron micrograph of an actual muscle, and this is what it actually is. If you look at one myosin, one heavy filament, you see 6 thin filaments arranged around it. And what happens is when the myosin contracts, the myosin heads bind to the actin, and they literally rotate. And as they rotate, the myosin rotates, and the heads literally bind to one actin after another. Okay, and that's how contraction takes place. If you can look at it in another way, there are 3 myosins per actin. Okay, in the arrangement. Okay. And each one is binding this actin and causing contraction in a different area of the thin filament, okay? So we went through calcium release in skeletal muscle in the first video. So you've got calcium released, and that actually results in contraction because when your muscle fiber is excited, your released calcium binds to troponin C. So you guys will recall troponin C binds calcium. And the troponin C causes a conformational change along with troponin I and troponin T. And they shove this troponin away from the actin the myosin binding site on actin, and they expose it so that now it can bind myosin. Okay, and you'll note there are the 3 monomers here, I, T, and C. The troponin I is different in skeletal muscle compared to cardiac muscle, which is why it's a nice specific test for myocardial infarction. If you get dead cardiac cells that are releasing troponin I, your test for troponin I will only detect the cardiac specific isoform of that. And there is a cardiac isoform of troponin T as well, and that is mostly found in cardiac muscle as well, which makes that fairly specific as well. You guys will recall. So I wanted to mention that in passing. That's a difference between the skeletal muscle and the cardiac, and it allows you to detect those troponins in the bloodstream and determine they help you determine whether the patient had a myocardial infarction or not, because they're different from skeletal muscle. And so in any case, your tropomyosin is now shoved away from your actin binding, your myosin binding site, and now myosin can bind to actin and form a bridge, which can and as the myosin head performs a power stroke, what you get contraction of the muscle as the myosin slides past the actin. So let's see how that works. Here we have a binding phase for the actin-myosin interaction, and the two bind in the presence of calcium. And then what you get is what's called a power stroke, where the head rotates and moves back. And so that moves the myosin this way, and the actin, or the myosin this way, and the actin moves this way. So the two move in opposition to each other, and that causes the ends of your filament to come together, and that's how you get contraction through this sliding filament mechanism. You then get detachment of the myosin head away from the actin, and then binding takes place again, and you start the cycle all over again, all right? And that's basically how this works. So to get a little bit more specific in terms of what's happening with the ATP and how this is all working at a molecular level, if we start out with the energized state, so this is myosin that has hydrolyzed ATP to ADP and phosphate. The ADP and ATP and phosphate are bound, and your myosin head is now cocked and ready to rotate, but it can't do that until it binds actin. So in the absence of calcium, your tropomyosin is covering up your myosin binding site on this actin, and the two can't interact, and so you just sit there with a cocked head, and nothing happens, okay? And you're not getting any contraction. This is resting muscle, okay? If calcium is present, it binds to troponin C, and along with troponin I and troponin T, that moves the tropomyosin away from the actin binding site. And now you get myosin binding. The myosin head, or the cross bridge, binds to the actin. And that results in a power stroke as ADP and phosphate are released, and this thing literally rotates and moves backward, and so it moves your actin this way and it moves your myosin this way, and you get contraction. You then get, note that in the absence of new ATP, this ADP and phosphate is released, and you end up attached, and that's where you sit. So this is called a Riger complex. Most of you guys have heard about rigor mortis, where the muscles are all stiff in a dead body. This is why the ATP is gone, and so your muscle is stuck in an attached state, and it's really stiff, and you're in and your muscle has formed a bunch of Riger complexes. Okay. However, in a normal muscle, you have ATP. It comes in and it binds, and as it binds, you get detachment of the myosin head away from the actin. Okay. And then you can start your cycle all over again, where your ATP is hydrolyzed to ADP and phosphate, and as that happens, your myosin head cocks again and gets ready to bind to actin again, assuming that calcium continues to be there. Okay? If calcium is taken away, as in during relaxation, so what happens is if release stops, calcium release from the sarcoplasmic reticulum stops, calcium is removed from the cytoplasm through the auspices of sarcoplasmic reticulum calcium pumps, or SR calcium ATPases, very similar to cardiac muscle. Again, different proteins, but they function the same. They take up calcium into the sarcoplasmic reticulum, and because that happens, cytoplasmic calcium falls, okay? And in white fibers, which we're going to talk about in a minute, the type 2 fibers, you actually have, in addition to that, you have a protein called parvalbumin, which actually binds calcium, and that helps bring calcium down as well and cause relaxation. It actually happens quicker in these fibers because of the presence of parvalbumin, bringing the calcium down and binding it. This is not in cardiac muscle. It does help remove calcium from the cytoplasm, free calcium. It brings the free calcium down more quickly. So if your calcium comes down, you enter this state where your myosin heads are cocked, but they got nowhere to go because the tropomyosin is covering the myosin binding sites on actin, because calcium is no longer bound to troponin C. And so what you get is relaxation at that point, where the muscle fiber is allowed to relax. It's not contracting anymore. Okay, so it's allowed to passively increase in length in response to perhaps another muscle that's contracting, as they probably taught you in anatomy. Okay, or in response to a load on the fiber, where the load is pulling the myosin and the actin away from each other. They slide back together, and there's no active connection, so they're allowed to just passively slide. So you get relaxation at that point, or at least a lack of contraction. Okay? A couple of more proteins now that we've talked about, the thick and thin filaments, that I want to very briefly mention here at the end of this video. This is the dystrophin glycan complex, okay? And it has dystrophin in it. And this complex connects actin through the membrane to the extracellular matrix. So this whole complex is anchored at actin on the inside of the cell, part of the cytoskeleton, and it's anchored in the extracellular matrix on the outside. So as you get contraction, you're pulling the cytoskeleton along, and what happens is this complex bears the load, and it pulls the extracellular matrix along with it. Okay? If you get disruption of this complex, you get what's called muscular dystrophy. Okay? As you might imagine, that causes a problem, because now the extracellular matrix is no longer carrying the load as this muscle fiber contracts. So what happens is your membrane tears instead, because it's carrying the load, and of course it can't carry much force. So you get micro tears in the membrane, it allows calcium to come in, and you get cell death. And that's what happens in muscular dystrophy. At least that is the predominant theory in the literature at the moment. All right, that's it for this video. In the next video, we'll talk about what alters force of contraction in skeletal muscle, and finish up our skeletal muscle physiology lesson.